Hello everyone. So today's topic is controlled drug delivery system, and today we will discuss about the terminology which is related to the controlled drug delivery systems and their definitions and also rationale. Okay, so let's start. With developing the novel drug delivery systems, or you can say with developing the controlled drug delivery system, some terminology also developed. Okay, some kind of like prolonged release drug delivery systems, rate control drug delivery systems, modified release, also included with the delayed release, extended release, sustained release, and also targeted drug delivery system. So, first of all, we can see the definitions of prolonged release dosage form or prolonged release, or you can say the extended release dosage forms. So, in case of prolonged drug delivery, there is no control on rate or site of release. It reduces the frequency and maintain the constant plasma level. Just reduce the frequency to maintain the constant plasma level. Here no control in case of rate, site of release, and also the rate. Okay, but in case of control or rate control drug delivery systems, here deliver the drug at a predetermined rate either by systematically or locally for a specific period of time. But here is a special term that is called targeted drug delivery systems, which is included the delivery of drug at a specific site. It may be a receptor site or disease tissue site. No drug goes to a non-target site. That means it is only for a specific tissue or specific receptor, or it may be specific organ. Next, another term that is delayed release preparations, that is enteric coated preparations, formaldehyde treated capsules. Drug release occurs after a lag period. That means in case of delayed release preparations, the drug release occurs after a lag periods. That means not the immediate, in sense of immediate, there is a phase. After that, the drug gives its therapeutic effect. This time period is known as lag period. Or in case of modified release doses, you can say doses delivers the drugs with a delay or after its administration, delays after its administration. That means when you administer the drug, after some times it start working. That is why this types of drugs are known as modified drugs, or you can say it is a special types of modification, that is delayed release. Okay, delayed modifications, or for a prolonged period of times, when it takes prolonged time to start their therapeutic effect, is called extended release. Okay, or or it may be a specific target in the body, that is targeted site. Okay, so let's discuss about most confusing terms, that is the sustained release and controlled release. So when we define a sustained release or sustained dosage forms, that maintains a drug release over a sustained period of time, but not at a constant rate. In case of controlled release, when we define the controlled release tablet or controlled release dosage forms, we can say controlled release maintains drug release over a sustained period at a nearly constant rate. That means in case of controlled release, there is a rate or site specification, right? But other hand, when we define a sustained release or sustained dosage form, there is no site or rate specification, but here time specifications is maintained, right? And next, number 2, difference between the sustained and controlled release. That's sustained release follows the first order kinetics. In other hand, controlled release follows the zero order kinetics. Let's some abbreviation regarding these terms and their meaning. Here is some abbreviation, most common abbreviations in which you can introduce with your in your hospital training or in your dispensary. So let's see what are the abbreviations. That is MR, which is indicated that is modified release, and SR, which is indicated as sustained release. SA, that is sustained action. XR also indicate extended release. ER also indicated extended release, and XT also indicated extended release. And next IR, that is stand for immediate release, and LA long acting, and DR also delayed release. So these are the most common abbreviation which can use. Let's see some common marketed drugs which are included with these abbreviations. That is glyciphage SR. It is the marketed or brand names of metformin hydrochloride tablets which is available in SR form, that is sustained release dosage form. And you can also see in here that is as for XT, it is a iron based tablet and which is available in XR form, that is extended release form. Okay. Also you can see in this picture that is Nemegesics IR. Here IM terms indicate that is immediate release. It is also a non-steroidal anti-inflammatory drug which is available in our market. And also you can see in this picture that is medicines which indicate the ER, that is also extended release. Osnagia 15 year, it is also available in our market, and formation is extended release. Okay. Next topics, that is rationale. So before that we have to know what are the criteria or what are the specification for a perfect dosage forms. So let's see what are the main things which should maintain for a perfect dosage form, right? So first thing that it should need, that is drug must achieve MEC to show therapeutic effect. So here is terms MEC, that is minimum effective concentration. When we apply or when we take some doses, it have to achieve its MEC, that is minimum therapeutic concentration. And next, that maintain consistency. That means avoid fluctuation concentration or fluctuation plasma concentration. In case of maintain consistency, you can say when we take a drug, its concentration should maintain a specific rate in our plasma, right? Next, required dosing intervals shorter than biological half-life of a particular drugs. That particular drug should maintain its dosing intervals shorter than, obviously shorter than the biological half-life of itself, right? And last, that is the patient compliances. It should be patient compliance. Before we discuss the rationale of controlled drug delivery systems, we have to know some limitation of controlled drug delivery systems, or sorry, conventional drug delivery systems. That is, absorptions is dependent on rate limiting, monitoring of fluctuations in plasma, frequent dosings, risks of overdose and underdose. On the basics of these limitations, we can compare it with a graph, and here it is that is concentration versus times in case of plasma concentration level of a conventional dosage form. So, first point or first limitations was absorption is dependent on rate limitings. As we all know, the conventional dosage forms is rate limiting, that is absorption, the drug is absorbed after disintegration and dissolutions which are involved within the rate limiting step. So, as you can see in this graph, here is the terms MEC and MTP. MEC, the concentration or minimum effective concentrations. By achieving this minimum effective concentration, drug gives its therapeutic effect. Okay, and that is MTC, maximum therapeutic concentrations. It indicates the maximum level of therapeutic effect of a dosage form, right? And the gap between the MTC and MEC indicate that therapeutic range, above which it indicate the toxic dose. That means therapeutics above the therapeutic range, it indicate it is a toxicity. Above below the therapeutic range, it indicate the underdosing or underdose concentration. And second limitations is monitoring of fractions in plasma. As you can see in this picture, there is a various concentration is present in case of conventional dosage form, right? So, sometimes you can see the graph is high levels and then deteriorated, or you can say in case of first times or first duration of times, the concentrations and times curves is proportional. After some times, it's become inversely proportional. The relation between the concentration and times is inversely proportional. First half it make a proportional relationships, and secondly after some times it makes a inversely proportional relation between the concentration and time. And here you can see the concentration of the plasma concentration variety is maximum. So, monitoring of this fluctuation is very difficult and need frequent dose. As you can see, the maximum concentration, that is C max, is for a limiting time or short time, right? So, to maintain the constant C max, you have to take this medicines multiple times, that is frequent dosing, okay? And that is risks of overdose and underdose. That means it may up the therapeutic range or under the therapeutic range. So, now see the rationale. What is the reason behind the controlled drug delivery system, especially that? So, first point, reduction of dosage frequency. By the controlled drug delivery systems, we can reduce the dosing frequency of a dosage form, particular dosage form. Next, second point, that is lesser drug exposure to biological environment. That means when we take a medicines or dose, it may affect in a minimum concentration. That means the plasma concentration level is low. Next, third, that is minimal blood plasma concentration fluctuations or only continue a concentration. Okay. Next, that is minimal adverse effects or side effects. Last, augmented drug delivery systems. Let us see some points which can easily point out our vision with the controlled release or novel drug delivery system. So, first point, that is release. In case of conventional dose delivery or conventional drug delivery systems, it is rapid and complete release within a small times. Within a small times, drug release its maximum level. In case of controlled release, they are slow, controlled after a time durations. That means release of drug is controlled manner. And next point, that is absorption. In case of conventional drug delivery systems, the absorption is poor. In case of controlled release, absorption is high. High absorption, high efficacy. So, controlled drug release is effective. Next, rate limiting step, which are included with dissolution and disgregations, which is maintained in case of conventional dosage form, in case of controlled drug delivery system or controlled drug delivery dosage form, drug release from a dosage form. Next, dosing. Frequent dose is needed in case of conventional dosage form. In case of control, there is less frequent manner of dosing. Next, patient compliances. Obviously, in case of comparison to the conventionals, control release patient compliances is high. Next, bioavailability, that is in case of conventional it is poor, control better. Next, plasma level. There is a very much fluctuations in conventional drug delivery system in case of plasma level concentration, but in case of controlled release the concentration are constant for a period of time. That means blast concentration levels of a drug is constant. So, let us see the graphs, control versus conventional. Here the blue dot indicate as a conventional dosage forms and red dotted or red lines is indicated as controlled release. As you can see here, it is a plasma level drug concentration versus time curve, where in case of conventional dosage forms we need multiple dosing to maintain a maximum therapeutic effect or Cmax. As you can see here, the concentrations of Cmax is different in case of multiple dosing, but in case of control there is a constant rate in plasma concentration. That means Cmax is constant in case of control release for a specific time period, but in case of conventional dosage forms there is Cmax is varies. Okay. Hope you understand that.